Good afternoon, everyone. My name is Jackie Parker, and I'm the Director of Affinity and Campus Engagement here at DeSales University. And I would like to welcome you all on behalf of Institutional Advancement to our Virtual Scholars Series, The Coronavirus and the Vaccine. What has become a continued developing pandemic, we are proud to present DeSales University faculty, Dr. Dia Beachboard and Dr. Joshua Slee, who will discuss the coronavirus life cycle and the efficacy and safety of currently approved vaccines. Today, I'm joined by my colleague and co-host, Dan Vellante.
Well, welcome everybody. My name is Dan Vellante. I'm a 2009 grad and the Director of Alumni here at DeSales. And I just want to welcome everybody here, let everybody know that there are a lot of alums who are tuning in both near and far. And our goal really right now is to just have meaningful programming for our alumni base, sales community. And we felt this was such a unique topic and such one that's so relevant right now that we're so grateful to have our DeSales faculty to give them a chance, not only to provide knowledge, but really to showcase what so many great people here at DeSales are doing and be able to share that with our community. Just a couple updates. Everybody currently is on mute. Nobody will be allowed to mute themselves throughout the event. So if somebody wants to ask a question, please feel free chat box. We'll also be live on Facebook. So anybody who is watching on Facebook, please use the comment section on Facebook to touch base with us. Back to you, Jackie.
Oh, thank you, Dan. I am honored to introduce today's presenters. Dr. Dia Beachboard is an assistant professor of biology here at DeSales University and teaches courses in microbiology, immunology, and virology. Dr. Beachboard has been studying coronavirus since 2008 and is interested in host pathogen interactions between coronaviruses and the host cell. Her research is currently focused on understanding how the coronaviruses evade the host's innate immune response. Dr. Beachboard has published peer-reviewed articles in bacteriology, coronavirus genetics, and cell biology, and innate immunity to RNA viruses, including the Zika virus. And joining her today will be Dr. Joshua Slee, who serves as an assistant professor of biology, the division head of sciences and mathematics, and the department chair of biology here at DeSales University. He has taught courses in cell biology, cell culture, genetics ethics, microbiology, and immunology. And Dr. Slee's research focuses on understanding the innate immune response, the involvement of the inflammatory response in various diseases, and finding creative ways to attenuate inflammation associated with medical issues. He has published peer-reviewed publications in the areas of exercise-induced asthma, cardiovascular disease, heparin signaling, and biomaterial rejection. So we ask you to please welcome our esteemed faculty members here at DeSales University, Dr. Dia Beachboard and Dr. Joshua Slee.
Thank you, Jackie and Dan. It's great to be here. I'm going to turn it over to Dr. Beachboard. She's going to start. I would just like to say thanks for coming, and I'm happy to see a lot of our current and former biology students tuning in. So welcome back, or welcome to our talk, and I'll let Dr. Beachboard take the stage to get us started.
Okay, and I would also like to thank everyone for joining us. So we are going to start today talking about coronaviruses in general and how they replicate. And that will be covered by me. And then I'll turn it over to Dr. Slee to talk about the vaccine. Okay, so in order to get everybody on the same page, what I wanted to start with is the components of a virus particle. And I will first state that viruses are not alive. And this is a very hot topic in we'll say virology as to whether viruses are alive or not. And I've lost this argument enough that I can definitively say they are not alive. So I've spent a good part of the last maybe 15 years trying to convince myself that they're alive, and have come to the conclusion that they are not alive. They cannot replicate without a host cell. So the components of the virus particle are: one, it has to have nucleic acid. So that is shown, just going to switch to a laser pointer. That's shown here with the black line. So that's the nucleic acid. That can be either DNA or RNA. And the other required component of the virus particle is this protein that protects that genomic information. So you have genomic information in the form of DNA or RNA, and you have a protein that protects it. Those are the minimal components of the virus particle. So some viruses have this third component, which is a lipid envelope or a membrane that protects all of that. Coronaviruses specifically will have this membrane. So now that we all know what are the components of that virus particle, and I would also like to point out that if you have taken any kind of classes where they talked about the basic components of a cell, this virus particle does not have all of those components, and that is why it's considered not alive. Okay, so I wanted to just briefly go to the central dogma. So this is taught like really early on in intro bio, that we have DNA, so that's going to encode every protein that we can possibly make in our body. It's going to be encoded by your DNA. When you want to make that protein, you don't want to have to scramble through all that information to get the right one, so you convert it into RNA. So you'll turn that DNA into RNA, and then that RNA becomes protein, and that will give you whatever functional component from the DNA. So I bring this up here because viruses basically look at the central dogma and are like, I don't like that. I don't want to follow your rules, so I'm going to do what I want. So viruses in general have several ways of disrupting this central dogma, but the one relevant to coronaviruses is they skip this step. They're like, nobody wants to waste time with DNA. We're going to start with RNA so that we can go from RNA directly into proteins. Because of this, they tend to have very small amounts of genetic material, so they can make very few proteins. Okay, so now that we're all kind of on this same page, I wanted to go over coronaviruses specifically. So coronaviruses are RNA viruses. So that is shown here in the black line in this virus particle. So they skip that DNA step. So there's never going to be a DNA step. They can infect a wide range of animal hosts, including mice, whales, cheetahs. Like if you can think of it, they can infect it. Bats are another one. There are lots and lots of coronaviruses that exist in bats. And then of course, they can also exist in us, so in humans. Okay, and so within humans, there are 7 known human coronaviruses, which I have listed down here. So there are 4 that cause upper respiratory infections. So that's just the common cold. And these have been around for over 50 years. And then there are 3 that are more recent that cause what's called acute respiratory distress syndrome, or ARDS for short. And those are SARS-CoV, MERS-CoV, and SARS-CoV-2. SARS-CoV-2 is the one that we are currently living through the pandemic of. So I will point out that SARS is severe acute respiratory syndrome. So what they've done is added severe and dropped the D, and it's the same as ARDS. So it's a severe form of this respiratory distress syndrome. So we have these cold viruses that have kind of been existing for a long time. And then we have these viruses that can also have kind of what we might call pandemic potential. So SARS-CoV-1 emerged in 2003, and this could have developed into a pandemic, except that public health measures really stopped this virus, and we have seen no infections with SARS-CoV-1 since 2004. So our public health measures, including wearing masks and washing your hands, completely stopped that virus. MERS-CoV emerged in 2012, 2013, and that actually is still circulating in the Middle East. This particular virus is spread from camels to humans. And when various researchers tested camels in the Middle East, the majority of those camels were infected. So when there is not a vaccine for those camels, we can't get rid of it in the camel population. There's no vaccine for humans for MERS, so we can't remove it from the human population. So that is still ongoing. You get cases of MERS. And then, of course, SARS-CoV-2 causes COVID-19, which is currently circulating. So I'm going to go back over to this virus particle over here. So we had our RNA in the black line. It has its protein that protects it, so that's shown in blue. It has this lipid envelope. Within that lipid envelope, there are a couple of viral proteins. So there's an envelope protein and a membrane protein. And then importantly, it has this protein in green coming off of that membrane, and that is called the spike protein. This is going to be the most important protein in terms of when we get to the vaccination section, because all of the vaccines that are on the market right now are using this protein. So again, spike protein exists right there. It's sticking out from the membrane. Okay. So before I go into replication, I just wanted to talk about the genome organization really quickly. So this is an RNA virus. I will again point out that there is no DNA at any step of this virus life cycle. And it encodes various different genes. So there are about 9 in the SARS-CoV genome and less than that in some of the other coronavirus genomes. So it has this very large gene at the beginning that encodes non-structural proteins. These are involved in virus replication. And these are the proteins that I normally focus on when I'm giving talks. But for the purposes of this talk about vaccines, we are going to almost entirely ignore these proteins. And then down here at the end of the genome, we have the structural and accessory proteins. So the structural proteins are literally the ones that I just described that are in this virus particle. And then the accessory genes are also involved in virus replication, and those can be different from one strain of coronavirus to another one. So that's usually where you get that variation in the number of genes. So again, the most important one that we are for this talk is the spike protein, which is right here. And again, it's going to be extended off of that membrane. So now I just wanted to go through the coronavirus life cycle, and we will start with our virus particle here. So the first thing that when the virus enters our body, it needs to be able to interact with our host cells. And for SARS-CoV-2, it is going to do this through the virus spike protein. Again, that's that green protein, is going to interact with the cellular receptor, ACE2. So you've probably heard in the news about spike proteins as well as ACE2, because those are the two components that allow the virus to get into our cells. So again, they're going to have to interact. So this virus can only infect cells that express ACE2, and that's going to allow for entry into the host cell. Sometimes that is through uptake in a vesicle, and sometimes that is through direct fusion with the membrane. However, it ultimately results in this virus particle releasing its RNA genome into the cytoplasm. So once you have the RNA genome in the cytoplasm, it has 2 main goals. You want to make more RNA. In order to do that, you need to do this early expression of these non-structural genes that I'm showing here. So those are the ones that are going to allow the virus to replicate more. And then later during infection, you're going to want to make these structural proteins. So again, these are the proteins that are forming that virus particle. So you're going to make, first you're going to make the non-structural proteins. This is going to help you, the virus, not you, expand its viral RNA. And then you're going to make the structural proteins so you can make more of those virus particles. So for coronaviruses, remember I told you they had that lipid envelope. They pick that up from our cell membranes. So they use specific cell membranes. You can see that these viral proteins are embedded in that membrane, and then the RNA comes in and it forms that vesicle around it. So once you do this, you get the virus particle inside what's called a secretory vesicle. So the virus is released from the cell without actually killing the cell. So you may have heard some viruses will actually kill cells as they release. That is not true for coronaviruses. And the last thing I want to point out about the spike protein before I leave this slide is that, so in this image, I'm showing you spike protein getting into those virus particles. However, this spike protein, it can get to the cell surface and be expressed on the cell surface instead of being incorporated into the virus particle. So this allows the protein to be seen by our cells in the body. So that's going to be important for whenever we talk about how our bodies see the spike protein when you get vaccinated. So that protein, most of it is going to get incorporated into the virions, but you can get expression of that spike protein that just goes to the cell surface and sits on that cell surface. Okay, I think I'm going to hand it over to Dr. Slee now, and he is going to talk specifically about, let me stop sharing. Oh, I got to get rid of this pointer real quick. And he's going to take over and talk about vaccines. While he's doing that, I am going to answer this question about which human cells contain ACE2 receptor protein. So several different cell types in our bodies express that ACE2 protein, the majority of which are in the respiratory system. So we have cells in our upper airway as well as down in the lungs that express those.
All right. Can everybody see my slides here? Just making sure. Quick thumbs up. Maybe. Yeah, you can see them. Thank you. First, I just want to say how excited I am that we have Dr. Beachboard on staff here at DeSales. She is a fantastic virologist. And it just so happened that fate happened that as we hired her, knowing she was a coronavirus expert, the pandemic happened to hit shortly thereafter. So we really got very lucky with her. So we're very pleased that she's part of our team. So I'm tasked with probably the more challenging topic, and that's convincing you all to get a vaccine, because I know there is some hesitancy out there. So what we're going to do now is talk about the 3 vaccines that have been approved through emergency use authorization by the FDA for the coronavirus, okay? And how I'm going to do it is really just start by looking at some pros and cons and then talk a little bit about the science behind them. All right, so the first type of vaccines that were approved were utilizing mRNA technology. So Dr. Beachboard did a great job talking about the Dogma. So we know now that this is kind of that intermediate step. And these are the Pfizer and the Moderna vaccines. All right, the pros is that they're greater than 90% effective at preventing you from getting sick at all. All right, so with these 2 vaccines, we see relatively few people that get sick. There are some breakthrough cases, but it's not that significant. And then the number that's never really reported is that these are almost 100% effective at preventing hospitalization or severe cases of COVID-19. So they work really, really well by vaccine standards. Very few of our vaccines are as effective as these are. There's increasing evidence of long-term immunity. So as these are emergency authorized for use, we only know as far out as we looked, and we've only looked out about 6 months at this point. However, it's likely much longer than the 6 months, but we have to wait until that data is collected. The nice thing about these 2 vaccines, and we'll talk about this in a little bit later, is they're easily modifiable. All right, so it's estimated that we will need booster shots with these 2 vaccines. And the nice thing is we can very easily modify the contents of these vaccines based on whichever strain of the virus we're afraid of or concerned about spreading the most at that point in time. However, there are a lot of cons that are associated with these vaccines. First of all, it requires a very, very cold supply chain. All right, so if you're in business or supply chain management, you know that if if it's cold, it costs more to ship. It requires, specialized trucks, specialized freezers, and those sorts of things. So it becomes difficult to transport. The other thing is those logistics are very, very complicated. It requires 2 shots spaced about 20 to 25 days apart. And because of that, as of today, about 5 million people that have received their first shot haven't actually received their second. They've skipped it. So about 8% of the people that have received their first shot never actually followed through and got their second. And that's concerning from an immunological standpoint because we don't know their immune levels after 1 shot. Okay. The other thing that I think shies people away from these technologies is it's new. Although mRNA technology has been around for decades, this is the first time it's been used in a vaccine. So I think that can be a little concerning to people. And anybody that's gotten these shots can attest, it does have some moderate side effects. So you will likely feel pretty crummy for a few days after receiving these shots, but that's actually a good sign that your immune system's doing what it's supposed to do. Okay. And again, as Dr. Beachboard mentioned, it all goes back to that spike protein. All right. That's what we're trying to produce in our bodies in order to allow protective immunity to form. All right. So we're really looking at this spike protein gene that will give us the spike to allow us to have an immune response. Traditionally, what you would receive in a vaccine is you would probably just receive either a heat-killed version of the virus or maybe a purified spike protein. All right. But in the quick development of this vaccine, we went a little bit different. And what was done was we took that mRNA for that spike protein, and that's what you're actually being injected with inside a lipid nanoparticle shell. All right, so it's a lipid particle shell with that spike protein gene on the inside. Okay. Now, again, Pfizer and Moderna, they're not the same company, so the exact sequence of those messenger RNA molecules are a little bit different, but at the end of the day, it's producing the same spike protein. All right, and then it is a 2-series shot, 21 to 28 days spaced out. The other shot that's been in the news most recently is the Johnson & Johnson shot. This strategy is a little bit different. It's an adenovirus-based strategy, which we'll talk about what that means on the next slide. But the pros here, the biggest pro is that it only requires 1 shot. As we mentioned, 5 million people never got their second shot, so that's concerning. Here, it's 1 shot and you're done. Gives you a much easier supply chain because it can be transferred and transported warm or not as cold. And we see effectiveness rates above 60%. So you might say, well, it's not 90%, but again, you have to remember that here we're looking at the efficacy of getting sick at all. So it's greater than 60% effective at you not getting sick at all. But again, if we look at that number about how effective is it at keeping you out of the hospital or keeping you out of the morgue, we see that it's close to 100% again. All right. And that's the number that the news doesn't really report, is how effective is it at preventing severe infection. And that's really what we want to with vaccines, is if they can keep us from getting sick at all, great. But the next best thing is keeping us from getting really sick. And because of that, then we have much easier logistics with that 1-shot system. The cons here, and I think there are a little bit more than with the mRNA technology, these aren't as easily modifiable. They're certainly easily modifiable, but not as easy as our mRNA vaccines. Again, it's a relatively new technology. To my knowledge, this is only the 2nd vaccine of this type that's been developed. The Ebola vaccine was just recently developed and is the first of its kind here, which is a separate whole other talk that'll benefit millions and millions of people on the earth. It does induce less robust immunity as the Pfizer and the Moderna shots. So that is a fact that it is not as effective. And then, as you probably all know from the news, it has been linked to blood clots. And it now carries a warning with it when the FDA then reinstalls or reinstitutes this shot. With the blood clots, the link is very, very, very low of a percentage. So women who use birth control or anyone who smokes are at a significantly higher risk of developing blood clots because of their smoking or their use of birth control. So the link to blood clots, while important to acknowledge and avoid those that might have a high risk of clotting, the risk is relatively small. And that's essentially what the the FDA team that reviewed this said is that the benefits of the shot significantly outweigh the risk of developing a blood clot. So how do they do this vaccine? Well, essentially what they're doing is they're taking DNA and they're putting it inside an adenovirus. An adenovirus is basically a common cold virus that isn't really all that infectious, isn't really that severe of a virus, and they kind of strip it down to its bare bolts, nuts and bolts, and then they put whatever the piece of nucleic acid material into it that they want delivered, okay? So when you get the J&J shot, you're actually getting a virus that contains the gene for that spike protein, okay? All right, so now we've had the 2 vaccines. What exactly happens after you get that shot in the arm, okay? Well, with Pfizer and Moderna, it's straightforward. So you're getting that messenger RNA in that nanoparticle lipid shell. Our cell membranes are composed of the same kind of chemistry in terms of lipid components. So it readily fuses with our cell membranes and dumps that mRNA right into our cytoplasm, all right? Once that mRNA is there, our cells do what they do normally. They translate that messenger RNA into protein. And those proteins then assemble into the spikes that we would see in the coronavirus, all right? And essentially through this vaccine, what we've done is we've taken our cells and turned them into COVID spike producing factories, okay? And that ends up what you get here. Let me get my pointer here so everybody can see it. Essentially then what happens is we have our cells that produce these spikes on their surface, and they also can produce them in these little immune complexes that display them on the surface of their cell as well, okay? Now, if we switch gears and look at the Johnson & Johnson shot, it requires a couple more steps, all right? Since we're injecting into your arm an adenovirus, that must first enter the cell through very similar means that Dr. Beachboard talked about earlier, all right? That then must inject the DNA into the nucleus, where it's transcribed back to messenger RNA. And once we have that messenger RNA in our cytoplasm, we see the very same process that we saw with the Moderna and the Pfizer shot, where that messenger RNA is translated to our spike proteins, and they're displayed on the surface of our cells to initiate that immune response, okay? So comparing it to a traditional vaccine, we're relying on our cells to produce this spike protein for our immune system to respond to. That's the big difference between these vaccines and other vaccines that are on the market that you may have received, okay? So then what happens after that process? So this really is the immune response to all vaccines, not just the COVID vaccines. But essentially what will happen is, as those cells die off, as our cells naturally do, some of that debris will be present in our bodies, all right? And some of that will be those spike proteins, all right? And our immune system has these things called antigen-presenting cells that go around and pick up stuff in our bodies that's really not supposed to be there, all right? And they will take that and present it to a type of immune cell known as a helper T cell, all right? And they're basically saying, here, look at this. Is this something that's supposed to be in this body or not, okay? And if this helper T cell says that's not supposed to be here, it'll then turn around and show it to a B cell, another type of immune cell in our bodies. All right, so this T cell, having received it from the antigen presenting cell, goes to the B cell and says, hey, I found this, I don't like it, what should I do with it? The B cell will interact with that T cell and say, oh yes, I see it on this cell here, which was a cell that was vaccinated, right? So this was a cell that received that mRNA vaccine. And it's gonna get those two signals to say, yes, that's not supposed to be here, we should start the immune response. And what it does is it starts secreting antibodies. And antibodies are one of the things that protect us from pathogens. All right, so when we have antibodies to a pathogen, and we happen to come in contact with that pathogen, those antibodies will bind to that protein on the surface of the SARS-CoV-2 virus, and through the process of neutralization, get rid of that virus, which essentially means we prevent it from interacting with our cells, so we eventually pass it out of our systems, okay? The other thing that's really cool that can happen is this antigen presenting cell can take that same piece of the spike and show it to what's called a killer T cell. Killer T cell sounds more fun. Most of the literature calls it a cytotoxic T cell, but killer T cell sounds more fun. So we'll call it a killer T cell. And it can actually kill infected cells. So ultimately with viruses, step one is, can we prevent it from getting into our cells in general? Step two would be, if we weren't successful in preventing infection, the next thing we have to do is kill off the infected cells. All right, so basically to save the higher organism or save the human being, we kill cells that are virally infected to get rid of them. And that's what these activated killer T cells do. Okay, so then lastly, we'll close with and have lots of times for question. The question then becomes, why should you vaccinate? And there's two main reasons why. The first is, you gain protective immunity without getting sick. All right, that's the best reason. If you're at all likely to contact the virus, which at the levels at which it's circulating in the state of Pennsylvania, you're likely to encounter the virus in your everyday life. Through these vaccines, you can be protected against it, and you've never had to ever get sick. Okay. The other reason is that you can protect those who are unvaccinated. And this is particularly important if you have elderly family members that you interact with, or if you have young children that are yet to be vaccinated, or if you have somebody in your family who's immunocompromised, maybe they're undergoing chemotherapy for cancer treatment, or maybe they're on immune modifying drugs for rheumatoid arthritis, for example. They typically aren't really in the vaccine category. So by all of us vaccinating, we can protect those that are more vulnerable in our population. All right. And really, whenever you teach an immunology class, this graph comes up. And essentially what we're trying to show here is, how does your body respond in time and magnitude to an infection? Okay, so let's say here, just for all extensive purposes, this would be the first time you encounter a pathogen. All right. Our innate immune system is activated right away. Our innate immune system is a very basic immune response that we have that's the same for anything that we encounter. All right, if you go out and step on a splinter versus whether you get COVID, the innate response is pretty much the same every single time. All right, and it's not very robust. But then what happens later is this adaptive response. That's those T and those B cells that we were talking about previously. Notice it takes about 7 days for that response to occur. Okay, and then you get a nice hefty response that can ultimately clear that infection. Okay, but then the next time that you see that pathogen, notice what happens. The innate response, it's the same thing every time, but that adaptive response happens almost at day 0 at the time of exposure, and you get a very, very robust response that can clear that infection very rapidly. All right, so what you could easily then do is just cross out antigen A, put in the vaccine, and say, I'm going to protect myself from ever having to even encounter this pathogen for real by getting a vaccine that's going to give me protective antibodies, so that the first time I am exposed to it, I have the antibodies necessary to prevent a severe infection. And then lastly, the other reason is to protect those that are most vulnerable in our population. All right, and you've probably heard this if you've been following the news. It's what we call herd immunity. All right, on the left, we have a population of susceptible individuals. Those are the, we'll call them blue on my screen. And we have infected individuals, which are red. All right, and this is what we saw very early on in the pandemic. Nobody's immune system had seen this virus, and it spread very rapidly through the population because there was no immunity. Okay. On the right is actually where we're hopefully headed in terms of our vaccine and those who have recovered from coronavirus. We now introduce people that are immune. All right, these are people that have been vaccinated or have had the infection, recovered, and developed protective immunity. And what we'll see, because there's a number of green individuals that are immune, this virus is not spreading rapidly through the population. And we even can protect a small number of people who can't be vaccinated for various reasons from the infection. All right, and herd immunity is the reason why you have mandatory vaccine policies in elementary schools, in hospitals, in certain college settings, et cetera, because we're trying to protect those that cannot be vaccinated. Okay. I know I went through that kind of fast. That's about 3 weeks of a lecture in a real immunology class, but we wanted to make sure we had lots of times for questions. So at this point, I think there are some questions. We'll go ahead and kind of take those now.
Great. Josh and Dia, I know that we're getting a lot of great questions, so please, everyone, continue to post them. One of the questions that everyone's talking about is kind of going back to the J&J shot. Can you talk anything about the booster, why, J&J method would not require a booster, and even about, boosters going forward for the other injections?
Let me take that one, Dia.
Sure.
So Johnson & Johnson's probably going to require a booster too. Most vaccines, very few of them do not require boosters. So if you think back to the vaccines that you've gotten throughout your life, most of the time you have to go back for boosters. And that's just because the way in which the body responds to vaccines, it isn't necessarily as robust as it responds to a natural infection. So if it's something like a heat-killed vaccine, that vaccine that virus can't replicate in your body, so it only activates one arm of the immune system or the other. So it really takes the best vaccine is a live attenuated vaccine that can replicate in your body because it activates both your T cells and your B cell response. I don't know if that answered the question, but boosters are likely going to be required for all of them as the virus kind of drifts in terms of which strains become more prevalent.
Awesome, thank you. And this goes here. How effective are the 3 vaccines with the South African and Brazilian and other variants?
I can take this one. So I cannot speak for Pfizer and Johnson & Johnson, but I did actually attend a talk last week where this was discussed for the Moderna vaccine. And for some of those variants, it is equally protective. Some it's reduced, but the antibody levels that are in your body for those viruses would still be enough to neutralize it. So they may be decreased effectiveness, but that is in cells experiment. In our bodies, it should be just as effective. However, as people are not being vaccinated and generating mutations within that virus, we could get strains that are further enough away that they are no longer protective, and that's whenever we're going to need those boosters so that we can maintain protection when that protein mutates enough that it can we're no longer immune to it.
Regarding the display of the spike protein on our own cells, what % of our cells display this? Are these eliminated as part of the immune response? And then they're also asking, do intestinal cells also have ACE2 receptors?
Intestinal cells do. Some intestinal cells have ACE2 receptors, and that's why you get some people who get GI symptoms of COVID-19 and not respiratory symptoms.
Thank you.
The first question.
So yeah, in regards to the first question, I don't think we know the percentage of cells that express them. I mean, you're injected with a very small volume in your arm. So, I mean, it's hard to say how many cells pick it up and express it. But it is a natural process that all of our cells would express proteins in this manner as a way of communicating with the immune system. So we're really just taking advantage of these processes that already exist and using them to our benefit.
And that would be, with the vaccine, it is a localized response. So most of the cells that are going to express are right there at the injection site, and you can have immune cells pick that up and transfer it to other parts of the body, but it's mostly local just to that site of infection.
And can either of you speak on Covaxin, please?
I'm only loosely familiar with it. I've been really following the 3 that have been approved in the United States. From my understanding, it shows pretty decent efficacy. It's like around 80% efficacy in the Indian trials that have been done. But that's about all I know at this point.
Maxine asks, with Pfizer clinical trials, there were a few people that developed side effects, Bell's palsy and appendicitis 6 months after the second shot. Did the J&J clinical trials have side effects months after the shot? Also, did Pfizer say what those individuals that experience these more severe side effects 6 months later in the clinical trials have in common?
So this is where I'll be honest with you, and it's difficult to link these things 6 months after a vaccine to the vaccine. So the CDC and the FDA do follow these through the vaccine monitoring system. And just one funny example of this, there was a doctor a number of years ago after receiving the MMR vaccine, submitted that he developed anger and aggression and turned green when he got mad. And this is legitimate. I'm not lying to you. He submitted to the vaccine, side effects that he turned into the Incredible Hulk after he got the vaccine. And it was only removed when the CDC contacted him and asked him to remove it. So you have to kind of take some of these with a grain of salt because it's 6 months after the fact, and they're self-reporting these symptoms. So yeah, there are some side effects. There's no way around it. But 6 months after the shot, it's hard for me to put those two things together.
And Juliet said a colleague of hers who lives in Canada said that she is getting her second vaccine dose in 4 months. Their philosophy is to get more first doses out first. Do we have any idea regarding the ramifications of this?
Okay, I'll take it. So we have a guess. So in the United States, technically you have to receive both because that's what the emergency authorization use was for. And since that's been approved, we've gone back and looked. And essentially what happens is they don't have as robust of immune response after one shot compared to two. So it's likely that they have some form of protective immunity, but it's nowhere near as high as it is if you've gotten both. The other problem is that if you wait out longer, you're increasing the length till that booster shot. We then don't know if you get up above 90% immunity after the booster shot. So really, I would disagree with what Canada's doing. I would say you have to follow the science, and the science says we need both within a certain set number of days.
Julie asks, if you have antibodies in your system because you had the virus and recovered without treatment, does getting the vaccine put too much stress on your immune system?
No, it doesn't. So generally speaking, if you say you start a new job and you work at a hospital and they require a panel of vaccines to work in that hospital, you really have two options. They can do your titers to see if you have antibodies, but more often than not, they just say, get the vaccines again. And we do that with lots of things. So with tetanus, diphtheria, and pertussis, if anybody's had a kid recently, they just say, we're just going to give it to you again. So there's really no evidence that it's going to overwhelm your system. And if you think about it, we're exposed to billions of bacteria and viruses on a daily basis just by living on the earth. Our immune systems are much more robust than we tend to think of.
This is related to the next question in the chat, which said, I tested positive 4 times in 2020 with no symptoms. Should I get vaccinated? 100% yes. So what that means is you are asymptomatically carrying the virus and shedding it to anyone you interact with. So what's happening is those times when you tested positive, you didn't develop immunity to it. So then you got it again. This vaccine is going to prevent you from getting it again so that you're not asymptomatically able to spread that virus to your friends and family that you interact with.
Thank you. Can you guys speak briefly on the difference between emergency use authorization and full FDA approval? And is there any risk with getting the vaccination without full FDA approval, particularly if severe side effects occur?
So essentially the difference is how much knowledge we have about the vaccine or the drug, for example, because we've done this in the past with pharmaceutical drugs. Really, it's just how much knowledge we have. And I would say if you're skeptical, we're probably going to see full FDA approval occur sometime this summer because of the number of people that have been vaccinated under the emergency authorized use. So we're getting to that point where the FDA will probably give it full approval. So at this point, I would say there's really nothing to be worried about. I would expect if there's any harsh side effects, we would have seen them by now, given that we vaccinated millions of people with these vaccines.
The next two questions actually kind of are things you've touched upon, which is how does my vaccine help a loved one? Is it because it get rid of the virus more quickly so they are not exposed to it as long? And also, is it possible to spread the virus at all if you've been vaccinated or have recovered from COVID?
So, I mean, I just addressed part of that with the person who tested positive multiple times. So there is increasing evidence that these COVID vaccines are preventing asymptomatic carriers. So preventing transmission from someone who has no symptoms to someone else. So that is how you are protecting your loved ones, is you are no longer asymptomatically shedding that virus. And so you wouldn't spread it to them.
And the more kind of global health perspective of that, the more people that we have vaccinated, the more likely we'll reach herd immunity, and the more likely we can all kind of go back to normal, and stop wearing masks and eating in restaurants and going to concerts and theater events and those sorts of things, and actually be able to be in the same room as other people. So that's really why, is the more people that are vaccinated and immune, the less likely those at-risk individuals are going to get sick.
All right, I think that's it for all the questions in the chat box. We do have a few more minutes if anybody else has anything to add in. I know trying to go through all of the questions, it looks like in the middle of most of them, there's a lot of thank yous and how extremely helpful today's session was. So just as a reminder, we will, and today's session is being recorded, we will then be able to put it on our YouTube channel. Everybody who had registered for the event will get a follow-up email with a link to the YouTube link. And we do have one more question that came through. We can finish it here. Could you talk about the safety and mRNA vaccines? You mentioned this in the first of you mentioned this is the first of its kind, but mRNA technology has been around for a while. Can you speak on this?
Yeah, certainly. So it is true that these are the first mRNA vaccines that we've had, and it's a different kind of vaccine strategy. So with that, we have to, understand that there is a little bit of risk anytime something new comes out. But as we progress in time, we are now realizing that these vaccines seem to be very safe. And it's my professional opinion that a lot of our vaccines are now going to go in this arena, because it's much easier to produce them. So I really do think that's probably where we're headed. And in terms of mRNA technology, what I was referring to there is scientists have been playing with messenger RNA for decades. We use it in research. We use it for all kinds of purposes. So we even do it here in labs at DeSales. We use RNAi to knock down the expression of genes using messenger RNA. So it's just something that we've been playing with for a long time and studying for a long time. Now it's just the first time that it's been introduced as a human treatment.
I completely agree. I think we are going to shift to that technology. So historically, when we inject these individual proteins into people, it actually is extremely costly to generate those proteins. So the advantage of this mRNA vaccine is we're actually allowing our cells to do all the work making that protein. So you don't have to generate mass quantities of these proteins. That is actually very expensive and can be potentially hard to make and distribute. So we're relying on the amazingness of our bodies to help us create this vaccine.
That's a great comment, Dia. So we have machines now that just can print DNA or RNA, just like a 3D printer can print objects. And that does revolutionize the treatments. The one question that didn't come in that I see circulating a lot that I wanted to address, especially for our college-age students, there is an erroneous link going around to infertility in women. That is completely false. So that was actually started by two prominent anti-vaxxers that allege that the mRNA sequence was close to the sequence of a protein that was important for the development of the placenta in females. And that's not true at all. I had our bioinformaticist, Dr. Barnett, doing a sequence alignment for me, and there's no sequence similarity to them at all. But at the same time, if infertility was a cause for concern in the vaccine, we would have to see that in COVID-19 itself, which you don't. So there's been no link to infertility among women who've had COVID-19. Now, you can say blood clots, we have a side effect with the Johnson & Johnson vaccine. That is likely true because COVID-19 causes blood clots to a significantly higher rate than the vaccine itself. So for those that are young women that are concerned about getting vaccinated, I wouldn't be. There's no evidence that suggests there's any link to fertility at all.
And just to add to that point, again, whenever I was at the talk last week that was talking about the Moderna vaccine, this question came up, and she actually said that during these clinical trials, they advise women to actively try to not get pregnant. And of the somewhere between 7,000 and 8,000 women who were actively not trying to get pregnant, there were 9 pregnancies during that time. So if it was causing infertility, those 9 babies would not exist.
We have a couple more questions coming through. If an immune compromised individual gets fully vaccinated, will the efficacy be compromised?
If an
Immunocompromised
I would say
individuals.
yes. If they're immunocompromised, that means they're not able to respond efficiently in some capacity in the immune system. So it would likely be that their protective immunity would probably be less than somebody who's not immunocompromised.
Yes.
And can you speak on where are we with the child vaccines? So 16 or under. Providers like CHOP are studying, right now. Secondly, do we know why kids seem to be immune to this?
So we're thinking the fall is probably a good time point for kids. CHOP is doing those trials now. The issue there is they have to dial in the dose. So kids generally get a lesser dose than adults do, or vice versa in some cases. So they're trying to figure that out, figure out the efficacy. And kids are not immune to COVID. We are seeing now that those under 18 are the prevalent group that's spreading COVID. So they're not. I think what happens is the kids are just less likely to get a severe reaction to it. Us older folks that are less healthy are probably likely to get it worse. But kids do spread it. You just may think they'd have the common cold instead. But I'm thinking hopefully by the fall that children will be eligible for the vaccine.
So for clarification, are you saying that the vaccine will stop someone from getting COVID at least for 6 months? I was under the impression that the vaccine is to stop people from getting severe symptoms that require hospitalization.
So remember, Dr. Slee told us that 90% of people won't get infected. Close to 100 won't get the severe disease. So for Moderna and Pfizer, I would say the vast majority of people will not get sick at all. Okay, I want to address this blood clot question because I think we missed that earlier, but it was like, how long are women susceptible to blood clots after vaccination? So in the cases that have been before, they have developed those blood clots 1 to 2 weeks post-vaccination. That's what we know about blood clots.
Okay. And Dr. Beachboard, are you able to discuss how high the mutation rate of SARS virus, like the flu, mutates every year and thus needing new flu vaccines? Do you see that becoming the case with COVID-19?
So the really, really cool thing about coronaviruses is that most RNA viruses aren't able to go back and say, oh, we introduced a mistake here, let's fix it. Coronaviruses have that protein that goes back and fixes it. So the mutation rate for coronaviruses is actually much, much lower than the mutation rate for things like flu. So it actually surprised me that we started getting all of these really prominent variants so early because we do have that ability to proofread. However, as people are, there's still a significant portion of the globe that is unvaccinated, and we get more and more people infected, more of those variants are going to come up. And I think that we will need boosters to those variants. I think that, with the example of SARS-1, we were able to completely control that virus, and literally we have not had infections since 2004. So we were able to completely clear that with public health measures. So if we can get people vaccinated, I think we can get ahead of that mutation rate in terms of vaccination and have it protected to where you don't have to get COVID-19 vaccines every year for the rest of our lives. Will that protect us from the next emerging coronavirus? Probably not. But with these mRNA vaccine technologies, I'm confident that we will be able to make vaccines to the next emerging coronavirus much, much faster. And we will have all of those safety protocols in place from this one so that we will worry about that when it happens.
And we have another question, which is probably one of our last questions. Have you seen thrombocytopenia as a side effect of the Pfizer vaccine? I believe it has to do with platelets.
I'm not aware of any. So it's thrombocytopenia, and I'm not aware of it with Pfizer or Moderna, but I could be wrong.
We'll do our last question just for the sake of time. What is the stronger immunity, the vaccine or having it had the actual virus?
Vaccine. Josh?
I'll give a different answer. So generally speaking, with most viruses and bacteria, you get a stronger immunity with actually getting sick and recovering. But the problem there is there's a risk, especially with COVID. It can kill you, as can the flu and as can other serious pathogens. So yeah, you might get stronger immunity, assuming you aren't one of those asymptomatic carriers that had a low level infection that just didn't give you immunity. So generally, yes, but why would you want to have to come down with the infection, risk serious health complications if you can prevent it with a vaccine? So I do agree with Dr. Beachboard that the vaccines in this case seem to provide better immunity because we have such a large proportion of the population that are asymptomatic carriers.
Additionally, there are reports of becoming reinfected with the virus, which suggests that your natural immunity from having the infection is not that good. Whereas with the vaccine, you are seeing that protection, which is why I picked vaccine. If you had asked me for not COVID-19, I may have given you a different response, just like Dr. Slee did. He gave you the very conservative response. I totally agree with everything he said. For COVID-19, vaccine.
Well, we would like to thank you both, Dr. Beachboard and Dr. Slee. As we close this presentation today, I just wanted to ask you, do you have any final thoughts or messages for everyone who attended today that you would like to share?
I would just say thanks for coming out and thanks for bringing an open mind and looking to be educated on something like this. Really, the future in terms of public health depends on all of us kind of being in this together. And I think that's an important thing to remember is it's a very Salesian thing, if you want to connect it back to DeSales, is we're all here to support one another. And to me, vaccines are an extension of that. Because if we don't and we focus on ourselves, we're not getting out of this anytime soon.
Agreed. And I will go ahead and give you the plea, please go get your vaccine. And if after this talk you are still uncomfortable being vaccinated, I'm happy to answer any questions. My email and my phone number is available on the website, and I would be happy to answer any additional questions you have that would make you feel more comfortable being vaccinated.
Well, thank you so much again to both of you for sharing your time, your expertise, and the research that you've done on the coronavirus. And we thank everyone who has tuned in today to join us in this presentation. We hope that you will join us for our future events as well. We have 2 upcoming events that we would like to invite you all to. On Thursday, May 13th, we have radicalization towards terrorist and violent extreme recruitment. And on Wednesday, May 19th, we have chemistry demonstrations program, the 20th anniversary premiere. So again, we thank you all for joining us today. And Dan, you want to share with us how they can watch today's episode as well as other episodes?
Yeah, so anybody who has a Facebook account, you can just go find the DeSales University alumni page. Everything was recorded and posted live, so it'll be there live basically forever. So you can always go on our Facebook page. It'll also be available on YouTube. And again, tomorrow, everybody will be getting an email for those who attended or didn't attend with a link directly to that YouTube page. And hopefully, you know, for all of these, this is our third episode of Virtual Scholars. All of them are on our YouTube channel and can be viewed at any time. Thank you and have a great day.
Thank you.